Good morning to one and all present here. I am Priyadashini Sarja from the Department of ISC, third semester. I'm delighted and honored to welcome everyone to this annual international conference on recent trends in healthcare innovation, AICRTHI-2024. It gives me immense pleasure to introduce our keynote person, Dr. Animesh Acharjee, sir, Assistant Professor in Integrative Analytics and AI, Department of Cancer and Genomic Sciences, University of Birmingham, UK. Sir did his undergraduate degree in electrical engineering from Northeastern Regional Institute of Technology, Itanagar, India, and master's in bioinformatics from the Institute of Bioinformatics and Applied Biotechnology, Bangalore. After his master's, he earned his PhD from Birmingham University, the Netherlands, on applied doctoral studies with Synergical Lion Cancer Center as biostatistician. Here, he extensively worked on big data analytics, cloud computing, and after his postdoctoral study, he was offered a scientist position with BASF Corp Design, Belgium. Before joining the University of Birmingham and Queen Elizabeth Hospital, he was working with University of Cambridge, UK, focusing on metabolic-driven diseases like obesity, type 2 diabetes, using high throughput technologies. His research interests include AI, integrative data analytics, predictive modeling, biomarker discovery, and methods for diagnostics. Throughout his career, he was offered many fellowships from the international journals, and actively collaborated with many universities. For example, Harvard University and the University of Cambridge. So far, he has published an astounding amount of 85 papers and H-index in his 25 based on Google Scholars. We are really honored to have you, sir.
Thank you very much for very warm introduction. I personally feel really privileged to here and talk about my work and research I've been doing. So just to kickstart the process, as you can see the title is a little bit very broad in my talk. So statistics, AI, and biology. I mean, I myself, you can see, I mean, just now she has told I'm not a clinical person, I'm not even a biologist, but I'm kind of blended all the things together nowadays. So I have multiple different affiliations, and then what those affiliations are talking to you. So as you can see here in this particular slide, some of them are in the translational medicine, some of them are cancer and genomic sciences, some of them are like a health data sciences. So you can imagine that many domains actually kind of merging together to solve a similar problem. So what I thought, I will give you a kind of a some of the examples, throughout in my group, what we are doing and how we are making innovation applications in the healthcare domain. Yeah. Now, one of the caution is that, I mean, I am going to talk about some jargons, so don't take it as if you don't know anything about the jargons, and don't feel like that, oh, I don't know that, and so maybe I don't know anything about that one. So don't feel like that, because I'll be going to talk about many different nomenclatures and new terminologies in this talk. Okay, so the first thing is that I would like to touch upon a little bit of the general introduction, and then I'll go through some of the basic concepts of the personalized medicine. Yeah, so personalized medicine is a new concept coming in, or precision medicine, people call it also precision medicine in the healthcare innovation. Yeah, and then, as I said, I'm going to talk through 3 to 4 different examples or the case studies. Those case studies are coming from multiple different domains, multiple different diseases, mostly on the cancer and gastrointestinal diseases. Yeah, so again, I'm working very much in the teams. There always a clinical person in my team who is giving me a lot of feedback about the disease progression or clinical aspects. Yeah, and me as a kind of a dry person in the sense, in a more methodological person, we try to figure out what can we do in this area. So before going into the detail, let me talk about my lab structure, how my lab structure is look like. So I kind of having 3 different kind of wings, you can say so. So first one is is about multi-omics or multimodal integrations, and then then what I do in this particular area is like looking into multimodality of the data set. So I'm going to introduce multimodality in a few minutes. Yeah, okay. That's my first wing, as you can see. So my portfolio. The second portfolio is the diagnostics. Yeah, so diagnostics meaning that in a times of the diseases, if we can develop some early indications about the disease. Yeah, so for example, sepsis. Yeah, so if you have a sepsis, you will die anyway. Yeah, so if you can identify this before 2 days or 1 week before, the medical people can take a lot of decision making criteria in that. Yeah. And the last one, which was the important one, is the development of methodology. So this is 100% more or less is about the statistics and computer science driven, and mostly computer science and like a cloud computing, infrastructure development, something on also AI, machine learning type of approaches, development, generative AI, interpretable AI, and all these things. I'm going to focus actually too much on this part because I believe because of the engineering aspects important, you also need to know what are the things you can apply using your knowledge. Okay, so so what is happening in the first two domains? So people, the students and the faculties interact with me. They have a very broad background. So people are coming from the biochemistry, people are coming from biomedicine background in these two portfolios. Okay, and the last one, I'm 100% looking for those who are the statistics background, those who are mathematicians, those are computer science background and very good at scripting. So for example, if I ask them develop a R-Shiny, they will automatically take scripts from the back end and do the R-Shiny graphical user interface and make the things very easy for the clinical people. Okay, so that's the introduction of my group, and the question is what we do day and day. Yeah, day and day we do the data, but data, how the data is flowing. How the data is flowing. There are three types of data in my lab flowing. Okay, it's very important how the data is need to be structured. If you don't know the data, you don't know the strategy. If you don't know the strategy, you cannot apply method. If you cannot apply method, it will be very difficult for you to sustain in the data-driven analysis. The first one is what I do. I collaborate with the people. Okay, I call it as a lab data. So I am not generating the data. My collaborators are generating the data. Okay, so they are saying, okay, I have the data. Can you help us? Yeah. Now, data is a very broad type of data. You don't have to worry too much about the nomenclature in this case. I mean, like the gene data, you can consider as a gene data. Metabolic data, you can consider as a kind of a protein data in a way. So these are very much depends on the questions they are asking. So the questions is not too much important for us in our group. The question is very important for them. Okay, for example, they want to understand the mechanistic aspect of the drug, how drug is applying to the biological systems, and then how drug is actually helping or not helping the diseases. And they ask some modeler like me to understand the process based on the data. Okay, so that's the one way of data. Second way of data, public data. Here, the data is already in the public. You just need to know where the data is and how can you use those data sets. Okay, novelty is not the data. Novelty here is the methodology. Okay, so like people I have seen, I was looking at the abstract. I mean, you can see the people used SVM, KNN, and lasso logistic regression, those type of methodology, and compared them and trying to understand specificity, sensitivity of the model, right? So here you can use the public data to understand which method is giving you the best. Yeah. If you consider area under curve, then area under curve, sensitivity or F1 score based on your criteria, right? So here, these are some of the examples, not extensively researched, but these are some of the examples. If you wanted to have a gene data, then it is TCGA, the Cancer Genome Atlas, is a resource for, I think, 2.5 petabyte, not even gigabyte, petabyte data. So can you imagine if someone telling me there is no data, I'll be doubting that how far you know about the data in the public resources. Yeah. Okay. Then the third one, which my affiliations with the hospital. So I spend 40% of my time with the hospital, Queen Elizabeth Hospital in Birmingham. So there we have a 2 million patients record, okay, electronic health record data. So data is consist of the following: the imaging data, ECG data, electronic cardiogram data, X-ray data, electronic health record data. Okay, so we call it as an unstructured data. Okay, so you need to use some kind of a NLP, natural language processing, and other methodologies to make it structure, and then you develop your models. Whereas these data sets are very much structure. Structure meaning that you have a defined rows and columns. Yeah. So you have a structure. Definition of the structure is rows and columns are fixed. Yeah. So these are a little bit different one, but what you get is quite a lot of the 2 million patients is just for hypertension. I mean, there are every day the clinical data we can have it. The problem is getting access to the hospital data is not easy in UK. Yeah, so we need to be clear 3 rounds of the security and all other things. We don't use AWS or Amazon Cloud because it is we have the proprietary web server. So the computations need to be web server. Data cannot be transferred. Someone need to be sitting in the hospital and doing the computation. Yeah, so you physically need to be present. We cannot even take in your home. It's a very, very high security data. Okay, so the two things, the key message from this slide, what I'm trying to tell you, that before you dig into the data, you need to understand the sources of the data, how the sources of the data coming in, what are the structure of the data, and then you go through the quality of the data, and then you develop the methodology, actually what you wanted to achieve from the data sets. Okay, so just because I have just told you the structure of my lab and the data, I think we are ready for going through the basic concepts. Okay, what are the basic concepts? The question you can ask yourself that, well, I mean, there are a lot of data. Is it happen randomly? Or is it why last 5-6 years people are talking about the data? Okay, data-driven decision, data analytics, data scientist, whatever you call it. I mean, is it a new domain or is it all together? It's kind of a evolve eventually. Okay, so what I did, I looked at back in the past, what happening. Yeah, so if you look in the horizon, if it's 1990 and then we are beyond to 2024, this one, what is happening here? And I'm talking about mainly on the biological domain. I mean is you can see the drug discovery. Drug discovery is a very tedious process. It takes 15 to 20 years, starting from the discovery to landed up a drug to be prescribed by the clinical persons. Yeah. And then the attrition rate is, I think 2% is a success rate. If you try 100 things, only two things may be going to be successful, and maybe out of 2 things, one is going to be successful if it is crossing through some of the legislations afterwards. Yeah. Okay, so what is happening in this before 2000 is driven by targeted application. What does it mean? People had a specific genes, people had a specific metabolite, and they used to look at these particular data sets, particular aspect. Okay, now around 2000 2001, you can see the star. A major breakthrough happened in the biology and the healthcare domain. What happened in 2000 2001? Human genome sequencing. Yeah, I don't know if you know it, but it is a big incident or big phenomena in terms of the mankind. Yeah, the human genome published. Yeah, so human genome started in United States government Department of Defense. It took 10 years of the project to finish and articulate this process. Yeah, in the parallel way, Celera Genomics started their human genome sequencing, and both the partners actually in the similar time frame they published those kind of human genome draft. Now, what happened as a consequences? Because human genome is a very kind of a nucleotide, ATGC. I mean, it's like a string theory. Those who study string theory, there is a kind of a string, ATGC, and you can see many billions of the data sets generated after human genome sequencing. Now, there were many problems in 2001. The problem number one, the traditional biology, those who are looking at the biology, they did not know how to manage those data. Yeah, and because there was what is the solution? So then they talk to the computer science people, those who have software engineering or computer science people, those who have already knew some sort of a basic SQL or the databases development. Okay, so then the first merge between the biology and computer science around 2001, and as a consequences, many databases came in. Okay, many databases came in, people trying to script too long in like a Perl. I when I was doing my masters, I did a lot of Perl programming for the scripting. Yeah. So people compare the human genome versus mouse genome versus other genomes and trying to understand what is the probability the two organisms are similar or not similar. Yeah, based on the sequences, if they can predict what will be the disease probability or risk of the person going to be disease. Yeah. Okay, so the people call it as a comparative genomics, comparing multiple genomes, and then approximately 2007, technology also evolved massively. Yeah, like high throughput experiments. Meaning, what do you mean by high throughput? High throughput means that you take one sample, maybe blood sample, maybe serum samples, or maybe mucosal samples, and then from the experiment you generate 1 million of measurements. Yeah, so it's not a traditional data, right? So 1 million measurement, thousands of measurements, it became a wide type of data. Yeah, wide set. So there are two concepts in the data science. One is the tall data versus wide data. Tall meaning that you have a less measurement, high number of patients or the cases. Yeah, that wide means you have a many measurements and a low number of the samples, and those who know already machine learning, we call it p greater than n problem in the machine learning settings. Yeah, all right. So there are a lot of problems in these kind of things, and then people said, okay, how each of the components of the molecular aspects we can combine together and make a disease prediction. Yeah, things change, evolved further down, and then people have trying to use the social network data like a Twitter or anything. If you tweet, can you do the sentiment analysis on the Twitter? What is the sentiment of the persons? These kind of things. And the nutrition, how much intake with the nutritions we are taking every day, how much exercise we are doing, can we measure in the real time? Yeah, so these are the questions coming in. As a consequence, it generated a very immense type of data set. Yeah, but also it gives you a multimodality. What does it mean? Nutrition data is not same as a social network data. So modality is different. Yeah, for example, electronic imaging modality is unstructured data, so modality is very different. So that's what we call it as a multimodal data sets. Yeah, all right. So what I'm going to talk about a little bit further is how we can measure those biological things. Okay, so just before going a little bit further, you have to understand, or you have to know, what is omics type of things. And I understand maybe you have not studied too much of the biological things in your curriculum, but what I'm trying to tell you, this is the central dogma of molecular biology, meaning that you have a DNA, RNA, and mRNA and proteins, and what is the missing is the feedback mechanism here. So what happening here is when the RNA is produced, either it is a self degrading, yeah, so it gives a feedback mechanism. Those who are studying electrical engineering, it is pretty much about the control theory. Okay, so if you can mimic a control theory, you have a control theory boxes, right? And then you have a input, so you can think about this is the input, it is a processing, it is an output. And usually it is not a CISO system, single input, single output system. It is a highly MIMO system, multiple input and multiple output systems. Yeah, I have simplified this process for understanding. Yeah, so eventually people have used actually control theory application like PID controller and other controllers to understand the biological system here. Now, here in this particular talk, I am talking about how can you measure it. Yeah, if DNA to RNA is produced, how much DNA is produced, how much mRNA, how much protein is produced. Can we measure those things, and what we need to measure? Yeah, and then eventually you can see here in the down there are many different types of the data actually generated based on only these principles. Okay, so if I wanted to capture, we need to do experiment, and these are the experimental tools. So if you talk someone in the biological science or maybe other universities, bioengineering, they will tell you the thing. They have done some kind of a DNA sequencing, they have done some kind of RNA sequencing, they have done MSMS, mass spectrometry. Yeah, I don't know if you have a chemistry in your first year of your curriculum, then you might have studied spectrometry. How can you do? And then you can quantify how much. Yeah. So what happened in this process is like in this biological process, you need to prepare the library of the samples, you need to do the quality control, you do statistical analysis, and then you get a biological knowledge. Okay, and then this need to be feedback in the person who are doing the biological science. Okay, and we call it as an upstream and downstream type of experiments. Yeah. So the key message, what I'm trying to tell you, you need a technology to generate the data. So how I do, I collaborate with the people. In my lab, I don't do any experiment. So I know the people who do experiment for me, and we collaborate. We write a combined grant together saying that, okay, maybe 50,000 is going for this experiment. This data will be coming to my group. My group will be helping for the experiment, modeling of those experimental data. Yeah. Okay. So I think it is very important to understand the where your niche area is looking at. You cannot do everything, of course, but you can do certainly many other things. All right. Okay. So the characteristics. Now talking about the characteristics of this particular data. What is happening in this particular data? As I said earlier, one patient, coming from sample from one patient, you get a multiple different measurements, usually thousands. Yeah, usually thousands. Not many of them will be relevant for your outcome variable. So what is your outcome variable? So outcome variable may be disease, non-disease. Yeah, outcome variable may be drug responded, not responded. Outcome variable could be the tumor size. So you can decide, is it a classification problem or regression problem around this type of thing. Yeah. Okay, so I have already talked about little bit the number of samples are less and variable, so the wide data, and then we call it as a large P, small n problem in the statistical machine learning theory. Yeah, there are many ways we can get rid of this type of problem. Yeah, so one of the other problem in this area is the traditional statistics. Traditional statistics is relying on too much on the assumptions, like a Gaussian distributions. If you're looking at any kind of a normal distributed graph, so that means you can use the t-statistics or linear regression. This kind of data doesn't have this kind of property. Yeah, and also you assumed there will be a lot of collinearity in your columns. Yeah, because of p greater than n, and because of this you cannot invert your variance covariance matrix. Yeah, so you need to use some kind of a pseudo solutions of your matrices. Yeah. Okay, so these are the things you kind of giving a little bit of idea what is happening in those data sets. Now, why I'm talking about those data? I'm talking about because I wanted to understand that each of the individual and their biological signals, how they're propagating. Yeah. What is the population, what is the precision medicine, and all these things. So what is the precision medicine? So if you look into this picture, you will not realize this is a population, right? This is the example of population, so like a crowd here, and you can see you cannot really differentiate between who is a control, or control means healthy person and the diseased person. Okay, now traditionally what people thought, in this particular crowd there are two types of classes exist. One is healthy, another is the disease. Okay, which obviously not true in that category. Yeah, because some of them will be not disease yet, but maybe, for example, think about the type 2 diabetes. I mean, type 2 diabetes, what we do if you go to the GP, or in this case as a kind of a private medical doctor, so they will ask you, okay, do you check your cholesterol, check your H1B1C, HB1C. HB1C is a marker for type 2 diabetes, and based on HB1C you can prescribe the doses of your metformin. Do you need to have a metformin or not, based on that. Yeah, so this is a traditional thought, but people are thinking no, it should not be like a black and white type of disease. It need to be something like, what is the probability of the person going to get a disease, or high risk in the disease, or low risk in the disease, or not at all disease. So it is not like a black and white color, or like a pink or green, blue color, but it should be some multiple different classes. Yeah, definitely multi-class problem, not a binary class problem in a way. Yeah. Okay, so in the personalized medicine and precision medicine, what people are assumed to achieve is that your population is a very heterogeneous population, is not like a black and white or like a healthy and disease. Population has a characteristics between the some people will be getting high risk to get any disease, yeah, or some of the people will be low risk of the getting disease, some of them will be within the risk. And how do you measure? We can look into the molecular profile of those patients, like DNA, mRNA, proteins. So that is telling you the biology. Yeah, that will tell you the biology, but then you need to be tied up with the technology. If you don't tie up with the technology, you cannot measure it. Yeah, I'm sure. I mean, in Bangalore or Karnataka, there will be many other, like IISc Bangalore, there are many other institutes. They do a lot of experiments, right? So if here, if it is engineering focus, you can help other people to collaborate with them and get the analysis done for them. Yeah, this type of approaches, and then you can see the matrices, and then what you all get, this type of matrices, end of the day, and then you have to analyze and making a data-driven decision. So data-driven decisions for whom? For unbiased estimates of the things you are looking into the matrices. Okay. Now the problem arises is that if you have all these molecular profile, how best we can integrate them and trying to understand the disease. Yeah. So that's called a fusion problem. We call it as a fusion or integration, because they are multimodal data. So some data is coming from image, some data is coming from NLP processing, some data is coming from, let's say, protein data. So how can you combine them? And that is definitely called as a integrative analytics. Okay, and that's how integrative analytics come into the picture in the scene. Okay, so what is integration? Yeah, what is integration? So if you look at the literature, you will find there are many different names. Some people call it a fusion, integration, multi-block problem. Yeah. And then, as you can see, right hand side, I just take this picture from, I think I have a reference in the later slide, showing some sort of histological slide, radiology, and then special transcripts and many other things. You can use it for many purposes: diagnosis, prognosis, survival, and side effect, many other purposes. But why this integration is necessary? Because if you integrate, maybe you can have, you can understand the mechanistics, you can understand the interactions between the molecules, and those interactions are very critical for your for the disease, and very important for further discovery in the process. Okay, so now I'm trying to give you some examples. So these are some of my papers. I mean, you can see the first one paper. This is my paper collaboration with Harvard Medical School, Department of and Center for Genomic Sciences. Someone called Taylor. Taylor contacted me, I think 5 years before, when I just moved into the Birmingham. And the reason Taylor got my contact is that because one of his employees was working with me in Cambridge, and we together looked at the stroke and metabolite. This was funded by one of the pharmaceutical companies, and together we worked some of the diagnostics aspects of those data, and we published it. There are other papers, like for example cancer cell paper. They talked about how you can integrate those multiple diverse data sets. You can see the multimodal, and these are many other very important papers actually. Cancer cell, Oxford paper, and then a couple of my paper as well. I mean it was when I was in Cambridge. I was working on the diabetes and other metabolic diseases, and we have published some important papers using the machine learning. We have used random forest machine learning algorithms using some kind of objective functions, so automatic feature selection procedure, and then trying to report it back. Yeah, and then this one is also a collaborative work together with Amsterdam Medical Center, working with the colon cancer and adenoma patients on the Dutch population, and trying to understand how those data sets are actually telling you. So these are a couple of examples. And then last one is talking about, this is a Cell Press paper, talked about how machine learning can, together with multiple different measurements, you can help with the phenotypic trait of your interest. Yeah. So these are quite interesting to know. These are some of them. Okay. So let's talk about what is the strategy. So I've talked about the data. If someone is giving you data, what do? You start analyzing? No, you should not start analyzing the data first. You should ask questions. What is the person bringing you data? What is the key question he or she wanted to answer? So if someone is giving you data and they don't know what to do, you should not start analyzing the data first. Yeah. So you just to set up a meeting and then trying to understand which system and all the things. So this is a very strategic slide. It tells you that if someone is giving you the data, multiple data sets, it can be different ways you can combine those data sets. Okay. One of the naive way of combination, like that you put everything on an Excel file, for example, if they measured multiple different things from the same patients. You can put everything in an Excel file and then trying to do, assuming this is a single data, and then trying to do your fancy methodologies there. That could be one of the way. We call it as an early integration in the literature. Okay. Second way is that you need to project the data in a high dimensional, like a kernel space, and then you calculate the in kernel space, and then you boil it down in your normal space. Yeah. So if someone has used support vector machine, then this is the kernel I'm talking about, maybe radial basis kernel, or maybe other kernel, polynomial kernel based on the functions, and then you calculate in a higher dimensions, and then you calculate and come back to the one. There are other methodologies like a principal component analysis or latent feature modeling. There are many other people have talked about these things in the literature. The last one, which is I'm using a lot and I have developed myself in the literature quite a lot. So I assume that each of the data structure is a different, so you analyze the data separately and then you combine them later on. So you can see a little bit of looking like some interactions, some of the node, yeah, so we call it node. Those who know the graph theory or the interactions of the networks, it's exactly the same the way the electrical networks or other networks are working. Yeah, it's coming from the graph theory algorithm, so you can use the DFS, BFS, all these kind of algorithms if it is a complex graph. In fact, you can also find out the statistical properties of the graphs, and there is someone called Barabasi, who was in the Notre Dame but moved into the Harvard Medical School. He has developed this field significantly in this way. So he call it as a topology of the graphs. Based on the topology, can you identify the structure of the diseases and other things? This is also a very active area of the research as well. Okay, now the thing is, I mean, you can further divide your late integration into two different parts. One is the quantitative integration and one is the qualitative integration. And those integrations are, I'm still working on the qualitative one, but I think quantitative integration is quite matured in the literature, and in my lab I were trying to contribute a lot in this. Okay, so let's talk about one of the quantitative integration in a way. Okay, so this is the cartoon, and this is published already. You can take these are my papers, all my papers, and as I said, last few years I've very actively in this domain, merging between the machine learning and graph theory algorithms and trying to understand the mechanistics between the biological phenomena, and then trying to talk with the clinical people, and then doing the experiments and validate it, and then report back to the literature. Yeah, so that gives very powerful. So your impact factor of journal 3 to 10. So if you don't do validation, impact factor stays in 5 and 6. If you do validation, impact factor goes 10, impact factor journal. Very respected journals. Yeah, so we trying to do some of the costing when you write a proposal in the project, so that we can validate those findings here. So what all I'm trying to tell you in this particular slide, you have a patient, you measure multiple different things, yeah, multiple different types of the data, multiple modality of the data. It gives you different matrices. That's what you got. Yeah, and then somehow you select your measurements which are important for your outcome variable. Yeah, so outcome variable could be case and control as a binary classifications, and if you use some feature selection algorithms on there like lasso elastic net or some sort of a ranking based algorithms, and then you can select saying that, okay, I don't need 100 of my measurements, I need maybe 5 or 10 measurements, and then I'll be happy about it. Yeah, once you kind of know which measurements you are going to forward in the pipeline, and then you do some sort of a graph theory based algorithms, and then trying to understand the mechanistics. Yeah, so you can see here the merge between multiple domains. I think it is a very important slide. Here the experimental people involved, here machine learning people involved, here the little bit of mathematical people involved, here again the biology people involved. Yeah, so it need to be very clear where you are. Are you here, where you are, where you are contributing? So if you are here, stick here, don't move around. If you are good at something, be good at something always. I mean, no, if you're moving too much, you'll be losing your time. By seeing your next your friend is doing something, oh, you're tempting, right? Because someone is getting like a 10 lakhs offer versus 15 lakhs because of TCS or some other, you say, oh, maybe I'll go for there. No, answer is absolutely no. Answer is where you are good at, you should do the where you are good. That's it. If you are good, doesn't matter. If you are here, UK, USA, doesn't matter. You are good as good. That's it. So that is very simple actually in terms of the things. Okay, great. So this is, I'm setting up all the stories. Now I'm going through the case studies now. Okay, now case studies tend to be a little bit of biological, so this is a caution. So and you don't have to worry for the biological problems comes from biology. We solve it as a predictive modeling. Yeah, and I'm sharing the same ethos. Yeah. Okay. Now there is a disease, and I never knew this disease when I was working. The first time I started working in this project, this disease name is called PSC, primary sclerosing cholangitis. Okay. So as I said, I'm not a clinical person, so I never knew about this disease. And when I talked with my team, this is a kind of a IBD type of thing, inflammatory bowel disease type of, yeah. And if you look at the literature, so what people have done, this is a Nature and Microbiology paper, and this is a Nature Review paper, which is a very high impact journal. Of course, I mean, Nature and Cell, I mean, what you else have in this literature? Lancet, of course, and New England Journal of Medicine. Okay, but this is a quite important topic because the clinical people cannot diagnose those things. Yeah. They mimic as a kind of inflammatory bowel disease, but they are not inflammatory bowel disease. So when the patient is going to the clinic, the clinical people tend to diagnose it as a IBD, but they are not IBD. That's what they wanted to understand, why there is a confusion in this type of symptoms. Symptoms look similar, but the diagnosis is very different. Yeah. All right. So what we have done to understand that, yeah. So you can see here, these patients are coming from Queen Elizabeth Hospital, where I am also spending some time. Only 10 patients. This is also a very rare disease. Just to keep in mind that one. So healthy control, only 10 patients we have. PSC UC we call it, and only 10 patients. Ulcerative colitis, UC for ulcerative colitis. So IBD has two types: ulcerative colitis or Crohn's disease. Together they call it as a IBD. Okay, so they have taken the mucosal samples. What they have done very, very deep experiments. Yeah, they have generated RNA sequence data, they have generated immunology data, they have generated the microbiome data sets. Yeah, I have not introduced microbiome, but I think I have slide here. I'll introduce a little bit later. Okay, but anyway, those terminologies may be for you alien word, but I understand that. But what you need to care about here, these data will be kind of a matrix format some point. Yeah, these data will be matrix point some point, and then here you use the machine learning algorithms to understand which marker actually impacting your disease outcome. Disease outcome meaning that your control patients versus your PSC patients, and then you do some kind of a predictive modeling. So that is very similar to the graph theory based approaches. Here we can understand what are the nodes, like for example, another example would be if it is very like aeroplane networks. Okay, so one of the hub is London Heathrow. Yeah, many plane passes through the Heathrow, so it is a hub, right? We also call it as a hub here as well, exactly same. Frankfurt hub, Delhi may be one hub, Mumbai is a hub. Yeah, so because many of the flight going for multiple different countries, so it is very important. Many connections, meaning that important ones. Yeah, all right. So first thing is that looking into the data. So I hope you know the some of the principal component analysis from the statistical knowledge, or some people nowadays call it as AI. I don't care what you call it, but my thing is that is it solving my problem, what I wanted to achieve. So if you do the principal component analysis on the data, so here you can see these are the patients, right? The green patients is very different from the red patients. That's what I wanted to see. Yeah, so meaning that and I have used 1200 measurement in my analysis, 1200 measurement to draw these figures. Yeah, so what all I am telling you the comparison between three different type of comparison. So one is a control versus healthy healthy patients, PSC versus ulcerative colitis, and then all of them are together. So you can see here this is very distinct separations between the control versus other diseases where it is not. Yeah, and the below this is the heat map showing if the genes are overexpressed. We call it as overexpress or down express. These are the terminologies we use in the literature. Yeah. Okay, and then if you do some kind of a univariate analysis, so if you do a t-test on those data, you can identify how many genes are kind of important for your analysis. Yeah, so I'll go a little bit quick here, and then also we can identify, these are all graph theory based approaches, we can identify what is the probability of those genes are associated with those particular pathways of your interest. Yeah. All right, so then we have done very similar analysis for inflammatory markers. We have done very similar analysis for the microbiome based analysis. And what is the methodology? So methodology is very simple. So when we wanted to understand the marker, so as you can see, in the x-axis in number of your features, those features are your measurements. Yeah, and you do a objective function of area under curve. You ask the program, or you script yourself saying that you want to have a minimum number of the features which gives you the best AUC. So this is you can literally convert as a optimization function in your model, and trying to understand how many features are important for that. Yeah. Okay, once you identify those, and then you put everything together. So this is the graph look like if you put all the data from the studies. Yeah, this is not like a massive network, but this is some extent very much non-interpretable. What does it mean? It means that you can see because of the many connections, some of the connections are negatively correlated, some of the connections are positively correlated, but then you see some of the very nice kind of a clusters of the things coming up here. And then question is, what does it mean? Does it mean something about your biological thing? Does it mean something about your system you are working with? Okay, so here we identified actually all the genes and all the things, actually the bile acid metabolisms and the involvement of measure of the genes, everything is up, and that's how the biology comes into the picture. So in this particular work, what I'm trying to show you, that you have your experimental thing, you do the generation of your data, you analyze the data, you understand the biology, and then trying to understand what is going to happen. So we got fascinated by this one, and then we have write a proposal to submission in the journal of, not journal, European Crohn and Colitis Foundation. Yeah, and then we found some of the grant money, that so those patients we have given a drug to understand if the drug is going to be responded for those particular patients. Yeah. All right. So, so this is the paper we published, and then this is a Journal of Crohn and Colitis, medical journal. But also similar type of methodology is also applied. I think this paper came out clinical cancer research, but on the pancreatic cancer. This is the collaborations with the University of Bern in Switzerland, and together with the Bern Hospital. So someone I'm working with called Eva, she is one of the leading pancreatic cancer specialists in the world, and we have published I think it is a 12 impact journal in the clinical cancer research. This is also a integrative analytics together with my work with University of Manitoba in Canada, and together with Vern Dolonsky group, and there we have looked at the Canadian population, the First Nation population, trying to understand how the integrative analysis has helped them to identify some of the key areas in the biology. Okay, and this is together with another department. We have done something on the diet and interactions between the older population versus the young population, and how the mechanism is going. So this is from the inflammation and aging department. So these are some of the examples. But if you look into the paper in the search, you will find that this is a very much interdisciplinary type of approaches. So I'm just looking for the time. Okay, so I have some time. Okay. Okay, so this is kind of a one part what I wanted to talk about, the integrative analysis. Now we are going to the diagnostics. Yeah, now diagnostics is very booming area. Yeah, why booming area? Some trillion, 1.2 trillion market. Yeah, if you can do anything in there, you are there. Okay, and there are many opportunities actually. I myself got surprised when I talk with my clinical people, is that they say there is no cure for these things. If you can save 2 days early, then we can save money, but also the patient experiment as well. That's why diagnostics is so important. Yeah. Okay. So I have started working in the diagnostics quite around 5-6 years before, and then in my group, what we look for 3 properties: robustness of the marker. Yeah. Predictive behavior. Is it a predictive? Yeah. Predictive means that if I do a trial in UK, if I get the data from India, is it, if I identify a marker in UK, is it the same marker is identified in Indian population? Yeah. So if not, it's not a biomarker. It's not a diagnostics. Diagnostics need to be across the globe. Doesn't matter where I am sampling my population, the effect would be same or similar. Yeah. Okay. Then the indicative properties. Indicative meaning that what does it indicate? This one. For example, HB1C is indicating for type 2 diabetes. Yeah. BRCA1, BRCA2 is a famous mutation for breast cancer. If you have a BRCA1, BRCA2, 99.5% sensitivity and specificity of those marker. Doesn't matter. So if you go to the trial in Africa or maybe in other countries, European population, the sensitivity specificity remains the same. Okay. So some of the examples. So these are the papers come from my own study. I mean, this is the editorial I wrote in the diagnosis, and how you can actually use AI for diagnostics. Yeah, and then a couple of other letter to the editor. I mean, so there are many ways you can publish your paper. Not only just it need to be just research work, but you can write a review paper. Number one, you can write to the letter to the editor. Okay, you can write to the reply to the paper published. If you have a very strong data, you don't agree with the paper who published. Yeah, and then you write to the letter to the chief editor, and then he will be happy or not happy about maybe not happy because you were replying to the published paper. Yeah, but anyway, these are the ways. So there are a couple of letters I have written because of the recent trend, and one of the trend, actually this one is very interesting. The reason, one of the Turkish group published a paper around I think it is 12 impact journal, and they have shown how AI based AI-based recommendations. Yeah. So now they have developed a recommender system. They have developed a recommender system based on the diet they are having in the patients. The patients who are suffering from gastrointestinal diseases. Yeah. And they have shown two different groups. One group, they are following the traditional diet intervention, and the other group, they are following the AI-based recommender system. And AI, they have used XGBoost. Okay, decision tree based algorithms in this study. Now, when I read this paper and I got very fascinated, and I thought this paper should be gone in a very other journals too. So I wrote to the editor, Therapeutics Advances in Gastroenterology, yeah, saying that okay, you need to take into account this paper a little bit, and then this is the upcoming domain, and then you need to publish this one. So I just wrote one-page letter, and this letter actually got published in this journal as well. And this is a 4.5 impact journal. So 4.5 impact journal, one page, why not? Yeah, and then if you think this is a, but the problem is you need to think where you are. Okay, you need to think where you are. Are you telling kind of a top-notch thing or not? And it takes a little bit of experience. All right, so I'm going to give you some of the examples I have done in my group. This is an excellent work done by Joshua Smith, and this paper is under review now, and the reason I'm talking about now, I'm going to talk about a lot of biostatistics and machine learning fusion in this particular work. So microbiome-based diagnostic in colon cancer patients, stratification and survival analysis and diagnosis. Okay, so Josh now in Oxford, working for CR UK Cancer, and other people. They have a collaborative, like Julian is from the France, and then Anissa and Anubrata in Glasgow, and Anissa is also one of my colleague, Department of Pharmacy here. Okay, so before I talk about a little bit more, we need to understand what is microbiome. I mean, have you heard about the microbiome first? Okay, so microbiome, you may not heard about microbiome. This is a game changer nowadays. I mean, a game changer. Why? Because the microbiomes are tiny. They live in our gut. Okay, they live in our gut. They could be the bacteria, they could be archaea, they could be fungi, they could be pathogen, they could be viruses and all the things. But they all are living in our gut. And some of them are living also in my oral. There are two types. One is the gut microbiome, and one is the oral microbiome. Yeah. Now, in the gut microbiome, what they do, they communicate with your brain by a gut-brain axis. Okay, so many of the neurodegenerative diseases, many of the gastrointestinal diseases, many of the cancer, their involvement is massive. Yeah. Okay, so if you like to know more, please read this paper. This paper came out in 2011 in Nature. They are talking about the gut microbiome is our next genome, because they are controlling so many things. They are involved in so many other aspects. Okay, all right. So they are kind of a foreign particles. That's why, and we are the host. Yeah. So we call, so host and microbiome. There is always a interactions. Yeah, we call it host microbiome interactions. Yeah, like yogurt, what we are eating? Bacteria. Yeah, probiotic bacteria. So if you eat yogurt, this is good for health. I mean, we knew earlier, but now people are telling in experimental evidences. You know, breast milk, everybody knows breast milk is good for the babies. But now people are telling, oh no, there is a gut microbiome is good for the healthy baby. Even after 20 years, they don't show up any disease. Some of the diseases don't show up if you give a breastfeed to the infant. So this is a very, very striking, and the last 10 years approximately, people are telling a lot about those things. Yeah. Okay, so I try to do something similar in a way. Now the motivation comes from those papers. So if you see, this is the Nature paper came out in earlier this year, in Nature Medicine paper, and then earlier 2-3 years before, there is a group from University of California, San Diego, the Rob Knight group, who is very actively working on the microbiome based profiling, and he wrote a review paper in Science. Yeah, and he was telling the microbiome actually is and what he did actually, he looked at 34 different cancer types, 34 major cancer types, and he found all these 34 cancer types, the microbiome diversity is massively impacted. Yeah, what is diversity? Good bacteria is dying, bad bacteria is taking over. That's the diversity we are talking about. Yeah. So of course, this paper came out, a very high impact one, and there are other roles in the cancer. There is another. This is last month actually published in very Cancer Research Journal, and people say, okay, the cancer research, there are massive impact on the colon cancer as well, the microbiomes. Okay, so So what I am asking in my group is, can microbiome diversity used to specific subtypes of the colon cancer patients? Yeah, meaning, what does it mean? It means that if you give me 100 patients of colon cancer and if you give me microbiome data, can I find a subgroup of the patients? Okay, so meaning if now I can translate this exact same question in my machine learning settings, do I have an outcome variable? No, I don't have any outcome variable. So what I'm telling is unsupervised learning, right? So given the data, can I find a clusters of my patients which can be different behavior in this data set? So this is a problem of unsupervised learning. So what we have done, and then further questions like which microbes are responsible, and the final question is, if the microbiomes are associated with the patient survival, what does it mean? It means that if you have a higher kind of a higher concentration of the microbes, maybe the patient is going to die anyway earlier. If it is a lower, so we predict it, some statistical modeling like Cox regression modeling and other things. Okay, so data collection, important thing. Where I get the data? So I collected the public data. Yeah, so remember in the previous slide I showed I use a lot of public data. That is the public data, TCGA data, and I have collected 585 samples, colon cancer samples, 1406 measurement of those microbes. Okay, and this is the references, and then I have used another data for validation. Okay, if you don't validate in a new population, and I am editor of multiple one, I don't even pass it to the editor. If any paper goes without validation, no value in the literature, because your data will be already overestimated. Yeah, doesn't matter if you tell me you have the tenfold cross validation. No, tenfold cross validation is just one way of doing it. Yeah, unless you have developed new methodologies. Yeah, so this is, we are very strict. Like I'm an editor of the Nature scientific data, we don't pass to the reviewer. Yeah, and because we know reviewer is going to ask you same questions to you. Yeah, all right, so validation important. Without validation, the work even preferable if you do experimental validation. If not, at least some of the data you show efficacy of your AUC or F1 score, how does it behave in your new data set. We are not interested to see the training data. We wanted to see the test data. We interested to see each of the fold you do, how predictability is going to happen. Yeah, so don't overfit the model. That's the message. Okay, great. So first thing, the data pre-processing, as you all know, the missing values and all the things, microbiomes, no missing value, clinical data, some normalization. So microbiome is a very different types of the data sets, so you need to take into the how you do the normalization. Yeah. So at some point you get some sort of looking like a kind of a quite normally distributed. Again, the right tail there is skewed in the right hand side. If you correct for that one, then it is ready for doing some further predictive analysis for your data sets. Okay, so then the first question, can microbiome diversity can be used? So what we have done many different algorithms. We have used Gaussian process, we have the K-means algorithm, KNN algorithm, you name it one, all the algorithms. Yeah, and then not taking the subjective decisions like K is equal to 2, 3, we kind of shown in a different categories of K how the class is going to happen in this case. So for example, elbow plot, silhouette plot, something like that. And then finally some kind of a decision based subtype identification, saying that what are the optimum number of clusters we wanted to get from these data sets. Okay, so we kind of convinced ourselves, saying that okay, this is the 3 clusters. Maybe we are looking at 3 different subtypes in the population. If you do some kind of a visualization in the 2-dimensional space using the PCA, using fuzzy means and palm matrices and K-means, it looks like that every patient, each algorithm has a preferences of the patient. So what does it mean? Group 1, if you do K-means, it is going to the group number 2. If you do another, it is going another group. So and also this is one problem. Second problem is that not all the clustering methodology is giving you the same result. Some of them are 2, 3. So what we have done, we have done a consensus sequencing, consensus clustering methodology. So we have used around 5, 6 different one. We took which one is mostly used in this process. Yeah. All right, and then this is something we check in the microbiome based world, that diversity, how the species level diversity is impacted. Okay, then the question is, so remember we have now 3 clusters, and then I'm asking the question that which microbes are important. Okay, so to achieve that, we have used random forest modeling, and we have used lasso algorithm, and then we have ranked those microbes, OTU for the microbes. So meaning that if you know how random forest works, the top one is most important, and we kind of optimized all the models, and then trying to make the data which is coming in both the models. Yeah. All right, and then the next question we are asking, okay, what is the probability those microbes are involved in the patient survival? So then we have done some Kaplan-Meier biostatistical modeling and Cox regression modeling, asking, okay, given these microbes, you can see this is one of the microbes are kind of the two lines are there. One is red one is less survival, and you can see this is low survival one, and then differences between these two curves are statistically significant as well. So we found only two of the microbes which can be, and then we have reported those in the literature. Okay, but this is just one part of my work, but then also that you can see these are in the diagnostics we have published. This paper came out in Gut. So Gut is quite high in terms of the gastroenterology. I think it is a 23 impact factor journal. And then the other paper is we came out together with Saraswati. Actually, together with you can see there is one name you might be recognized, Professor Anappa. So I'm working with Professor Anappa, and I am the second supervisor of Saraswati. And she published in biology, and the other papers are also talk about how you can use the data for diagnostics purpose. But diagnostics, I mean the data have not seen, meaning that validation results. Validation results are more important. All right. So any questions so far? No? All right. Okay. So I think I'm going a little bit more on the method development side. Okay, what we're doing in the method development. So method development is maybe very interesting. It's very much computer science driven and very much mathematical driven in this. So, but problems are some extent we are also kind of trying to solve those problems ourselves, but in the clinical settings. So the focus of the development is development of workflows, graphical user interface, Shiny, for example, and how you can make the life easier for other people. So there are multiple other methods like generative AI, explainable AI, and all the things. So just to give you one example, this is work done by one of my student. He developed Power Tools. Power Tools, what it does, it compare multiple different versions of random forest modeling, feature selection process, and then it gives you an estimate how many samples you need for future study. Okay, so he developed a Shiny interface. I think it is already cited around 40 times last 2 years work. So it is quite, but also that someone came to me 3-4 years before, and then she developed a workflow including fuzzy logic and other clustering methodologies, and we published this paper in this particular journal. Okay, so now I'll go into a little bit of some other very deep examples where I wanted to show some explainable AI kind of thing. So explainable AI is the booming in terms of the clinical settings, because if you do the deep learning models, if I ask you, okay, you have done deep learning model, tell me which features, which measurements you came important in your layer, you kind of looking at and then say, okay, so deep learnings and other models are good for prediction, but not for interpretable. How can you make interpretable thing? That's the other thing. And another part of the deep learning models is that they need a massive number of the patients. They need a massive number of population. If you tell me you have used deep learning 20 samples, well, I know now what happened in this model. So it is not going to be passing anywhere. 20 samples, you don't use deep learning model. So you need to it's like bringing elephant in the room and you cannot manage it. Okay. So this is a work from Serena. Serena was my master student, University of Birmingham in Dubai. And I am lucky to happen that I am also the deputy program director of health data science in Dubai. So I go to Dubai, teach and come back in 2.5 weeks. It's a very intensive course. But then she contacted me. She wanted to do something on the diet. She's I think because of her family members or other things has some other sentiment associated, and then she worked this is her master thesis I published in Cells iScience paper. So if you want to publish, I mean it in 3 months you cannot do anything unless both the people are working parallel. Yeah, this is very clear. Last 6 years, I am publishing those people working with me for master students. And this is the very proud moment. Even if I go for interview, they ask me what is proud. That's the proud moment. PhD people, the master student working 3 months getting a paper. So meaning that if they apply for abroad, like other countries, their CV will be like top 3 anyway. So they will get a call at least for the PhDs. Okay, so let me talk about what happened in this particular data. So these data, I have access to the UK Biobank data. UK Biobank data is millions of the patients, yeah, and diversified measurement. You can almost get what you want anyway. Okay, so we got a license here. Okay, so we have used 2 data sets. One is the plasma, metabolites, and diet, 2,676 patients, and we have validated these models in another data sets. Yeah, that's the validation cohort I'm talking about here. And then we have used some predictive modeling using interpretable AI, and then we also looked at the diet and metabolite interactions, how the interactions are going to happen in this portfolio. Okay. So data extraction. So first step is the extracting the data. So you need to get a little bit of understanding about how data came from. It's not that someone is giving you data and don't know how the source. Because if you don't know the source, your analysis will be incomplete. You need to take into account some of the variation in the sources. Yeah. So one of the way to do it is that you include inclusion and exclusion criteria of your patients. Yeah. So we get approximately like some of the food questionnaire, those diet are as a like people are asking, are you eating meat, are you eating egg, or these type of things, have you eaten, so these are the questionnaire form, survey kind of data. Yeah. Okay, so and then also we have done the same for the validation. So this is my training data, this is my validation data, very clear. Yeah. So the idea is develop something on the training data, predict on the new data set. Yeah, that's what we do in the predictive analytics. Yeah. All right. So what we have done in the data modeling. So I'm showing you the results now. You can see here. This is a multiple different models. So we have used a random forest. We have a Lasso. We have used XGBoost. We have used a light SBM, light GBM, and other things. And we have shown there are some kind of a signal here. But you can see the plasma are not very good. You can see AUC value is 0.67, which is not great in terms of the AUC sensitivity and specificity. In the contrary, if you can see here, this data is quite good. Like you can see the 0.92 AUC value in the comparison. Yeah. All right. But the thing is that we need some kind of an interpretability. Yeah. So for the interpretations or interpretability, we have done the SHARP-based modeling. Yeah. SHARP is one way you can do some kind of a more interpretable. What does it mean? It means that if you look at this, comparing the UK Biobank, you can see the global importance. So glycan A may be very important, but you don't know which patients actually high abundance of the glycan A. So we can have, this is a kind of a red color settings, which is showing the high concentration and low concentration anyway. So we have embedded this one, but also we have looked at a little bit of interactions in a kind of a UK Biobank in IBD cohort and non-IBD cohort, and this work is entirely on the IBD. Yeah. So finally, we have concluded that some of the metabolites coming from the fecal samples, which are more predictive compared to the plasma samples. Some of the key features we found fecal nicotinic and metabolite, nicotinic acid and metabolites, and diet metabolite associations we found stronger in the IBD population. Yeah. So we have published this one, and yeah, so it was really great, and now I'm following up with this, and I'm taking other measurements and trying to make a very comprehensive analysis with like proteins and the other clinical features. Okay, so let's move on. So next one is the generative AI. So you have known the LLM and ChatGPT, right? So I mean, if you don't know, then you are not in this world anymore. I mean, kind of. I mean, everybody knows nowadays if you are doing the computer science. Yeah. So the question is, how can you use generative AI in this type of medical domain? One is getting the recommender system or something like that, but can you use this embedded? Now, one of the problem in okay, before that, I need to introduce Sam. Sam was an excellent student working with me, and he is now doing PhD. Oh, by the way, Serena is doing PhD in DKFZ in Germany. She was in Dubai, but she is now in Germany doing the PhD in German Cancer Research Institute. Sam was working with me. Now he is a PhD student at University of Birmingham in another department. Yeah, so he did an excellent. Again, this is another example of master thesis. Yeah, so. We wanted to work with the generative AI, but then one of the major problem of generative AI is this one. not generative AI, the class. So usually it happens if you have a low number of the sample in one class. Let's say you have like a fraud and non-fraud, yeah, in the banking category. So if you have a fraud, let's say 20 persons, 20 people in fraud, yeah, and then you got a non-fraud, 5,000. So and you develop model, what is going to happen? 20 people. So definitely there is the model will be overflowed by the 5,000 individuals, right? If you don't take into account. Yeah, so it need to be taken into account, and here comes generative AI applications. Yeah, so that's what he did here. But in a kind of a omics type of, in terms of a omics type of biological data, we have a very similar issue in the biological data. So class 1 is a number of patients or number of samples are quite high. Class 2, the patients is very low. So the idea is, can you make it bigger like this? Yeah, and then you do the analysis. Yeah, okay. Now class imbalance, the state of the art, what is happening in class imbalance, and this is a problem of the predictive analytics domain here. So there are two ways you can handle it. Yeah, you use statistical modeling, you use the algorithmic approach. The statistical modeling, you can do oversampling, you can do undersampling, you can do SMOTE, the people are using it. Yeah, this is very well known one. Or you do the variational autoencoder, or you can do the GAN, generative AI, generative adversarial modeling, or you combine generative adversarial modeling network with a VAE. Okay, there are many approaches, but then both the domain has got a pros and cons. So you need to survive with that. Yeah, so either you choose one or other one, but then it has to be kind of a, it's not like everything is green. There is a kind of a lot of things in these areas. Okay, so what we have done in this particular one, so just to show in the left hand side, first you can see we have used the transfer learning methodology. So we have simulated the data. We have simulated the data in a very the high impact one. Yeah, and multiple different data sets. Any questions? Okay, so we have used multiple different simulations, and then simulations we have shown if you use the transfer learning based approaches, and then you can compare those ones. So in the simulation settings, we have used actually all these different comparison, like expand GAN, SMOTE, RO is for random oversampling, and all those methodologies we have compared, and we have shown one of them is quite the expand GAN actually performing better compared to the other GANs, but the improvement was not very significant. So we're trying to improve nowadays in a multiple different other modeling strategies. Okay, and this is the last example I am going to tell. This is about the feature selection algorithms done by my PhD student Laura, and this is the high dimensional statistics. So the question is that the data was given to us is like a longitudinal data. So the patients within 1 hour they do the measurement, within 24 hours they do another measurement, in 48 hours they another measurement, same patient, longitudinal, repetitive sampling. Yeah, so they wanted to understand if the dynamics of the patients in each of the time frame and can it be useful for other purposes. Yeah, so we have looked at the mod is for multiple organ dysfunction. So for example, if you get a sepsis, you get a MOD, so sequential failure of the organ. So if you are taking drugs or something, so if your liver is damaged, then initially within the 12 hours maybe there is a high probability that your kidney going to be damaged. Yeah, this kind of phenomena is called a multi-organ dysfunction, but we wanted to predict what is the possibility based on some measurements is a multi-organ dysfunction can be impacted. Now this paper also came out in Nature Scientific Data, is a very high impact journal as well. So so together me and Laura worked very hard on this particular project, and then what we have shown here, these are again the pipeline based approaches. So you give the input data, you measure the quality, we have used ElasticNet and Lasso kind of algorithms, but then we have modified it, not just as it is. So that's the innovation, we made in the algorithms, and trying to identify the markers and then clinical relevance of those markers. Okay, so if you look at the mathematical aspects, you can see that this is sum of square, residual sum of square, and then we have penalized those measurements which are not doing anything in this model. Okay, and one of the important thing what we have done here is generating one of the distribution, random distribution, and another distribution, permuted distributions, and we have shown AUC is significantly different or not. Yeah, so this is a very high dimensional based statistics, but we could able to show that in each of the dynamics, each of the time frames, some of the markers actually up and down significant. So you can see here, this in time frame 1, this particular marker measurements is very highly significant, but then it gone. Time 2, something else is coming up, and then time 3, someone else is coming up, other signals are gone. Yeah, so that gives us a feeling that if you target drug for particular one, you cannot target for other one. Yeah, so it need to be quite important for other aspects. Okay, so I have talked a lot about those ones, but there are many other papers. If you search my name, you will find it. We have done quite a lot of work, including bioinformatics, including graph theory based classifications and algorithms, where we can contribute as well as a engineering background. Yeah, so first of all, what I have shown that is an integration. It helps us to understand the mechanistics of the disease. Mechanism is quite important. It gives you therapeutic target, where to target. 20,000 measurement, if it is 1 is important, or 2 important, 3 important thing, that gives you target validation. So you need to do some kind of validation. Yeah, this is very important. And how I do it, I collaborate with the people who are very good at doing the experiments. Yeah, and selecting features which you reduce the dimensionality, priority. Method is very important. So if you know one method, don't tend to use this is the only method exists. Yeah, it doesn't work like that. I mean, I have seen people, when I talk with them, they have something in their mind and they want me to use that. Well, I don't work like that. I let data decide method rather than you decide method. Yeah, so I'm very clear in my communication. I'm not going to use that. I will rather, data will decide which method will be optimum, so which needs to use multiple method and then decide it. So the first important communication is very important in the first part, and making expectation clear. So I tell that I am not going to use that what you are saying, because you are not working in your domain. You are a clinical person. How do you know what the health data science is going to happen? So this is very important. One size doesn't fit at all. I have seen many times. Yeah, and your probability of fail is more if you are doing that. Okay. Interdisciplinary research. I think it is very clear that I myself came from the engineering background and working with the clinical domain. What I ever learned on the team spirit, right? I mean, I never knew all these things. So it's a combination. Data driven, hypothesis free. You don't do any hypothesis. Let data derive your hypothesis. Yeah, and also the little bit of the clinical driven, like they knows what, and they need to be marriage between these two domains, and this is happening. Believe me, this is happening to some extent. Yeah, okay, but everything I said, there is a lot of confounding factor. Gender, I mean, if you have 2 females in the group and there is a 50 male, gender makes a differences, and we have many papers are there like that. Gender, BMI, age group. If you are sampling like less than 20, and suddenly some of the more than 65 years old patients are there, that makes a difference and act as a confounding factors. We can correct them, so it is very important to correct those confounding factors as well. All right, so collaborations and interdisciplinary. So I do collaborate with many different, as you can see here, within Birmingham, but also my portfolio actually other countries, very actively working. So when I say very actively, I really mean it. It's not like a talking and then we'll do it 5 years later. No, it doesn't work. I don't work like that. So if you really want it to work, it need to be a little bit concrete, planning, strategy, goals, where we're going, and then some target. Without target, everything is like a fluffy. So this doesn't work in research. Okay, all right. And then finally, I wrote a paper to the editor, chief editor, saying that how these things need to be done, because some of the concepts are very new. Some people are coming from engineering, some people are coming from medical domain. What will be your leadership aspect? What will be your project management? How is it traditional project management versus your leadership? Do you believe in leadership as a traditional leadership? So I argued the leadership need to be changed. Leadership need to be changed, and I developed this framework. Okay, so your leadership in these 2 circles need to come out from your center. Okay, and then you need to communicate with other domains as well, equally important. So if you think what you are doing important, other person will think the same. So you need to be communicating at the same level, not even less. If other person thinks you are talking in a less, nothing is going to happen. Yeah, and we have seen it many times. All right. So it is very important. I talk about it wherever I go, and I think it was coming up quite nicely in these studies, and because of the human nature and the biology and marriage between data science, statistics, mathematics, and computer science. And this is my group. So my group is again very heterogeneous. I mean, in my group there are people who are very much expert in computer science. If I ask them to develop a GUI, they will say, okay, 2 days it will be ready. And they will ask me, is your backend script ready? I said, yes, backend script is ready. It's just front end. Okay. So, and then there are people like very good at biochemistry. They understand the disease, clinical people. Yeah. So I'd like to acknowledge my funding bodies, which is Health Data Research UK. I would like to also take opportunity to acknowledge Hypermarker, which is 10 million funding we got 11 countries, and I am leading from UK the diagnostic research, and Welcome Leap funding from United States, 6 million grant, and I am also leading from the data analytics, and other group members. They have contributed significantly, especially the master students. They come with a vision, and they like initial time is like they don't know what to do, but then after 3 months they get their paper. That I think it is the most amazing thing in my career, that helping those students and then working parallel. No, I don't talk. I mean, I will sit with you, I will see the scripts. That's how I do, and I made my environment around me, everything is like proactive. Yeah, they don't, I mean, so it worked very, very well. Yeah, and I'll be happy to connect with you, and happy to be collaborate. And yeah, if you have any questions, I'm happy to answer. Any questions? Very good question. Okay, I think I have a lot of discussion yesterday with Professor Ravi sir and other people. Okay, I will tell you now. So my current collaborations with Professor Anappa, yeah, I try to connect with Amrita. I try to connect with other MIT in Delhi, and I must say there is something which is lagging behind. I don't know if it is me, but I'm Harvard people want to connect with me. I don't know what is it. I don't want to blame anyone, but I don't know what is it. Somewhere, something lagging behind, and I need to figure out what is it. I think you will know it better. That's the people across the world. So yeah, I mean, I'll be very happy. As I said, I mean, I'm very proactive in this. I mean, and I would like to take the next step.
I understand that the way in which you are doing it, really proactive. Yeah. Very nice. We need people like you.
Yeah, thank you, sir. Thank you very much.
Sir, my name is Anil Kumar. So, very happy to listen to your talk, sir. So, I have a very simple question. When the change in technology from databases to data science and machine learning and AI, and the computing speed increased in a couple of years. Then how do you see the future of healthcare, sir?
Yeah, it's a very broad question and very good question. So in the future healthcare, I think what is going to happen, lot of clinical decision support system is going to be made. Yeah, so those so remember, I'm saying not clinical, they will not make a clinical decision, but they will support your decision. Yeah, so many AI and algorithms, those will be embedded on those kind of a decision supports. Yeah, I think today one of the ma'am was talking about digital twin kind of thing, and those kind of things are going to happen more and more. Yeah, many things will be cloud based, kind of a centralized. I don't know how much we can do in India, because in India one of the problem is the population, and we need to centralize everything. In UK, the way we are doing it is in the healthcare is 100% centralized. Like if I go even in Europe, even so, if you have got any disease, any part of the UK, or if you are in European countries, there's some extent they can access your data to see this one. So that is going to happen in more and more. I mean, the people who have not taken it, they are going to take it more and more. That's one thing is going to happen. Second thing is going to happen, as you said, the speed, the retrieving. the data. Yeah? If the patient is sitting and the retrieving of the data is taking quite a lot of time, it's not going to happen. So the speed matters a lot. Yeah. Second, then the capability of the storage. Yeah, how we can make the capabilities of the storage more and more. Now, Amazon and other providers, they made it possible some extent, but I think it is going to happen more and more flexible around those lines. So on the fly, the storage and also the archiving of the data as well. Yeah. The one problem is going to happen, which we are facing in the UK system, is the authority of the data. So if the patient is coming, are you authorized to use the data? So we are lagging behind, like protocols. You need to be place a lot of protocols if the patient want to use their data for the research purpose, and that is 100% lagging. That is one part, and bias in the data. Yeah, we need to correct those things, but other things will improve. Bias meaning that, for example, if the patients are coming, we kind of like minority ethnic group also need to be taken into account some extent, so which we are not doing very good at the moment. Yeah, so these are the holes in there, but then future healthcare systems will be more and more interdisciplinary. So like the computer science or IT, in future I envision there will be like a health component to apply, like a banking. Yeah, like banking is a big sector, insurance is a big sector for driving your policies, and they call it as a data-driven policies. So these things is going to happen more and more based on the data, but it doesn't mean it is going to replace someone who knows the domain expertise. Domain expertise will be still there just to support. They will support your decision, not take away your job in a way. Yeah, sure.
You said actually to develop these medicines, earlier they were taking 10 to 15 years.
Yeah.
But during this COVID, we developed the vaccines in a very short span of time. And how was that being done?
Yeah, so the thing is, yeah, that is true. The traditional way of looking at the drug development is 15 to 20 years. That is very true. But also we have seen in the COVID times. Now, COVID times was little bit different from the other systematic ways. Now, COVID, we knew which viruses we're targeting. There is a different versions. That's why every year you have to take the vaccine to protect from the H1N1 or something like that. Some different varieties. Yeah, every year they change it. But one of the good thing, I mean, not so good, but we know the target. These are the virus. So we identify the structure of the virus, which help massively in the drug discovery for the vaccination process. Yeah, but there is no drug. So it is a vaccine. Remember, this is not a drug. If you take the drug, discovery is still a lengthy process, but probably for the COVID, it will not take 15 years, maybe less than that, just because we got a vaccine and targets are well defined. Yes, sir.
I think probably I would like to say, sir, for the intervention. Basically, I'm from Mysore. I can speak Kannada well. With your permission.
Yes, please.
With your senior teacher's permission. My name is Dr. Basavaraj Madhusudan. You could see me in Google, and I have traveled extensively all over the world. I spent about a couple of years in UK. Next to Birmingham, I was in Institute of Food Research. I was working on carbohydrates, and I moved on to United States of America. I was there about in 4 different universities, about 12 years of service in research. My areas of expertise is biochemistry, nanotechnology, including food science and all. Okay, with this background, I would like to just interview with your questions. You know that, see, metabolomics, there is a omic technology, is there omics, different omics are there, which is closely related to the metabolic system. It is interested in the genomic system also. And nowadays the AI is coming up, machine learning is coming up. Now they are integrated with the biological systems. So that is the reason, in case if you want to continue in your areas of research, you may be from electronic engineering, electrical engineering, or the computer science, or whatever may be the region, even in civil engineering also, that could be linked to nowadays biology. Now it's an integrated science, it's coming up. So I am locally available. I am basically I am residing in Utagalli. I'm very close to your principal, Sadashiva. And so you could talk to me anytime if you are interested in a little bit of this tagging with your computer science or information technology with the biochemistry, biochemical aspect. You please consult me. Later on, you could take my number. You could speak to you, and I will speak to your senior teacher. So we teachers, we will discuss about it, and we will respond or will incline to the student interest, and there will be ample of opportunities to leave the country and go abroad. And now it is integrated science and engineering and with the medical science. You can go anywhere else. You may be a civil engineer, you can get into the medical biology also. So that, with that, you please, you could meet me later on, and in continuation with this topic, I would like to just address you. Okay, thank you so much for your patience.
Thank you, sir. Any more questions? Yes, I can see one hand in the back side.
So good afternoon, sir.
Hello.
Sir, my question is that as we are a student right now pursuing engineering and seeing after so many domains, and if we want to start some research papers or anything like that, would you like to guide on that? How can we start and what would be the procedures and all? And my another question is like, if we want an opportunity to do some research in some foreign universities, like as your university, like University of Birmingham. So what are the chances and how can we apply, and please guide on that.
Yeah, yeah. Okay. So this is again a little bit broad kind of question. So first, I think if I remember, first you were asking how to do research or something. So it starts with basic questions. I mean, for example, what you want to do. I mean, if maybe you were second year, third year, or fourth year of your curriculum. I mean, you have seen maybe some of the things fascinated you. It could be your algorithmic design or maybe some of the database design. So I think the first thing coming from there. So you wanted to pursue a little bit further career on this particular domain. That's one thing. Second, the next question would be, if you want to do, who can provide you the guidance? Who can kind of take it? So here, I mean, from your college, there, I'm sure there will be many people, many professors who will be knowing the process. Yeah. So that would be your first contact. Okay. And then based on that, if you wanted to pursue your further career, for example, masters, and you wanted to focus on that in the foreign universities, so you need to see which are the universities are actually providing those. Yeah. I would say don't go via name of the university. You go via your interest. I mean, I have seen people, they go via the universities. The problem is, university is great already. How does it, I mean, you went there when you were not getting what you wanted. Yeah. So you drive your thing where you can grow your own thing in a kind of your own interest. And then you might want to contact that particular person. Yeah, in that university, foreign universities. If it is a master's, then there will be a master education department. You can there is a universal email address. You can kind of contact them, and they will come back to you what you need. Like they usually need, for example, I'm involved in many of the education thing. Usually they need your GPA, your IELTS program. I think I don't, and maybe TOEFL and IELTS. If you are looking for United States, the TOEFL and IELTS. 6.5 for UK, I think for my university 6.5 for IELTS. They need, if you wanted to apply for fellowship, there is a deadline. If you wanted to, I think there are 5-10 fellowships they will provide you. Yeah, or if you don't get it, then probably you need to be self-sustained. Yeah. So this is the process. Okay, if you are already doing masters, trying to do PhDs, then you definitely need to find a niche area. And I believe that if you're doing masters in particular domain and you will be writing your thesis, the thesis would be your very close to your things you want to do in future. And then you look it for the people who are working in your domain. and connect with them. And I would highly suggest if you can publish your master's paper. I mean, the people working with me, they come with the planning. For me, I mean, I am kind of an organized person. So if someone is like LinkedIn or other things, many people contact me. So the first thing I ask, what is your interest? And maybe I like to talk with them. And I ask them, okay, look at my profile. What are the things you would like to do? Okay, if you can tell me you don't like anything, that's fine. Yeah, I'm not like, oh, I'll be offended. It's not offended. I mean, I cannot do everything, of course. If you like anything, then I can help you. Otherwise, it will be wastage of your time. Yeah. So, and then you choose one area, and I ask them to write a proposal with the timelines and other things. Yeah. The reason I want to put the timelines, if it is open, then it keeps open. So it's like a 1 week, next week. So I don't want to have, I want to avoid that. I don't do it. I don't want my members, I don't encourage my members also do the same thing. Yeah. So it is very targeted. And then I kind of work with them and then trying to see how they progress. I involve other people too. If I see the person is very promising, what I do, I actually introduce them to my network. Like one of the person wanted to do a diet-based modeling. And her interest is only diet, so nothing else. So okay, so I have a collaborations in University of Parma in Italy. So I have introduced them because they are doing a lot of diet profiling, the molecular profiling of the diet, like polyphenols, yeah, all these things. So I believe that they can learn more also with interacting with them. Yeah, but then the initial time frame, you need to kind of go through this kind of process, and it is everyone. If someone is applying for PhD, I do the same. If someone is doing for postdoc, I do the same. And there are many students actually just contacting me like they got many papers. So you need to drive it. I mean, I may not be doing everything because of the busy schedule, but as long as you have a target and you wanted to do where, yeah, we can help there. All right. Any more question? Okay. Yeah.
Good afternoon, sir.
Yeah.
Good afternoon, sir.
Yeah. Hello.
I'm Akash. As you mentioned about quantitative integration, so in that you mentioned you're going to take the attributes separately and create a model for them and then combine them. And like how, on what basis would you combine them together, and how efficient would it be than taking all together and creating a model?
Good question. So, this is the differentiation between the early integration versus late integration, right? So, in the late integration, as I said, we identify the marker or measurement, yeah, which is important, and then later on there are many ways to combine them. The one of the combination is like just do a simple correlation. Yeah, so correlation X versus Y, you can get some interactions. Okay, now, and then I check two things here. One, I check the interactions and I check the combined modeling, joint distribution of those data. So now it becomes the joint distribution because you have the measurement for data 1, data 2, data 3, let's say 3 data sets. And then some of the measurements are existing from data 1 and data 2, data 3. Yeah. So if you develop a predictive modeling on the data 1 with very specific measurements, comparing all the measurements are combined, you see usually the improvement in your predictability. Okay, so for example, if you do a joint probability distributions based on your data 1, data 2, data 3, they gives you a little bit better AUC. That's what we have shown actually. I mean, if you look at some of my papers, you will see that using a single data versus combination of the data gives better predictability. Now, how it will be different? It could be a little bit different comparing earlier. Yeah, because the interactions remains not the same. Yeah, so in the earlier case, if you have a 1000 measurement and then data 1 as a 1000 measurement, data 2 have a 500 measurements, in those cases what will happen, some of the unimportant thing will be interacting and coming up in your selection process. Okay, which is not wrong some extent. So that's why the two different approaches, if you do in parallel, you can compare what is the complementarity between two approaches. But more or less you will capture the real players in the sense who are actually impacting your outcome variable. Yeah, so that is quite important in this area. Yeah. Okay, there is another question here.
Good afternoon, sir.
Right.
Sir, I am Mahadev Swami. I am working as a faculty here. I need to know how exactly our institute can collaborate with your university? What is the procedure?
Yeah, so there are many. I think I was talking to Ravi sir yesterday about that. So there are many different programs in the faculty members. So there is like I mean, in particular, my group, what I want is that you write a proposal first of all, and because we need a visa process. Visa process is very bureaucratic nowadays. It takes quite a lot of time, and in the visa application also, not visa application, but then to get a visa, we need a document. Yeah, they want us the proposal as well. Yeah, so usually 6 to 8 months prior you arrive to the UK, and you identify any of the faculties in UK, and then based on that, what you do, you contact with them, regular contacts, and then and identify what are the areas you want to focus. Some people focus on the development, some focus analysis, or some on modeling, and once you identify the focus, then what can be done? You submit it, and then through the faculty members, and then if it is me, then I have to pass or sign off to the other departments. Those departments will take it, and then will check how you are going to sustain yourself. Yeah. So usually for faculty and internship, we cannot unfortunately provide any kind of a financial benefit unless you are postdoctoral fellow. Okay. Now we have seen the postdoctoral fellow usually come from 2 to 3 years time frame. Yeah. So you'll be considered as a kind of a part of the faculty, but not as faculty with limited time. So that is another way of doing it, but it is very competitive. Yeah, so it comes for 2 years and a 2-year salary and other things, and it comes usually from the grant. But these type of programs, like spending 3 months or 6 months time and getting some idea and coming back and implementing those ideas, that doesn't give any financial. So you need to look for maybe the institute, like the college, maybe support you for your FDP, faculty development program. Yeah, for 3 months. Yeah, and then what I have to do is submit your applications and proposal to another department. They will carry over these proposals based on that, and there will be around, as I said, 6 to 8 months time before you arrive. So that is the kind of a rough procedure, but this is one way. Another way is that there are some fellowships sometimes, University of Birmingham and UK based fellowships. There is a faculty exchange program as well. Yeah, so you can apply for that, like Royal Society. Royal Society is a funding body. You can apply for Royal Society. Royal Society is very generous. British Council, yeah, those kind of funding bodies are very generous. That if your a proposal is very strong, they will support you. In fact, they will support you 100%, more or less. Yeah, so including flight tickets and all the things they will provide you. But as I said, they're very competitive. They would like to see what are the work you have done earlier. They would like to see how much alignment the person you are working with, how much alignment it is done. I mean, if it is someone, for example, I don't do any database work, right? So if someone is database and they look my profile, oh, this guy doesn't do database. So why it is? So they will reject immediately. Yeah, so I think it is very important. The person you are choosing, some extent, need to be aligned. But also there should be some extent you need to learn as well. It's not 100% expectation. Yeah, so yeah, I think, and if you write me in specific questions, and I can definitely come back to you. Yeah. Yeah. Okay, so I think there is no more questions, so I think we can conclude. Yeah. All right. So thanks a lot for all the interactions, and really enjoyed the interacting. Yeah.
Good afternoon, one and all. I would like to take this moment to express my heartfelt gratitude to our exceptional keynote speaker, Dr. Animesh Acharjee, sir. Thank you, sir. Your thought-provoking presentation on this topic and teaching us that engineering and medicine can go hand in hand has really made us motivated and inspired. I also would like to thank our distinguished guests, our teachers and students who have joined us here, bringing energy and curiosity to this event. I thank the teaching and non-teaching staff for their support and their curiosity. Thank you, everyone.